Alright, so we're going to begin talking about proteins. Alright, so the basic building block of your proteins is going to be your amino acids. Now, every single amino acid has the same simple structure. Alright, so you have a carboxyl group, you have an amino group, you have a hydrogen, you have a central carbon. Alright, that's what makes up the backbone of every single amino acid. Alright, now how do the 20 naturally occurring amino acids vary from one another? Well, that has to do with this guy here, your R group. Alright, your R group is what varies, and that's how you get different amino acids with different properties and behaviors. Alright, so if you will recall that your carboxyl group and your amino group can function as an acid, and your amino group can function as a base. Now in the body, alright, at pH of the blood, your amino acids are going to be ionized. Alright, so your carboxyl group has a negative charge and your amino group has a positive charge. Alright, so that means that the beginning of your protein, your very first amino acid, has a free amino group with a positive charge, and the last amino acid that makes up the protein has a free carboxyl group with a negative charge. Now, let's look at the different types of amino acids. Now, I'm not going to hold you responsible for memorizing the amino acids and what group they fall under and what they look like. Alright? I would give you something very similar to this table on an exam if I wanted you to use that information to answer a question. Okay? Now, looking at your amino acids, alright, you have your nonpolar amino acids. Alright, so here you have your simplest amino acid. You have glycine. Alright, so your R group is actually a hydrogen. Alright, now notice that the rest of these R groups, they're mainly made up of hydrocarbons. Alright, so your hydrocarbons. Recall that your hydrocarbons are nonpolar, and that portion of the molecule makes that amino acid nonpolar. Now, your polar amino acids. Here, if you look at, for instance, serine, you have your carbon atom attached to an oxygen and a hydrogen. Right? Well, that oxygen is highly electronegative, right? Alright, so you have a polar R group. Alright, again, oxygen, hydrogen, polar R group. Oxygen, hydrogen, polar R group. Nitrogen, oxygen, polar R group. Again, polar R group. Remember that oxygen and nitrogen are more highly electronegative than carbon or hydrogen. Alright, for this instance. And then you have your charged R groups. Alright, so you have your acids and your bases. Alright, so your acids are going to have a negatively charged R group. So something like aspartate and glutamate. And then your basic R groups, your basic amino acids, are going to have a positively charged R group, like lysine, arginine, histidine. Alright, now understanding that amino acids are either nonpolar, polar, or charged. That tells you a lot about how those particular amino acids behave and interact in a protein molecule. Alright, so talking about protein synthesis. Alright, so here, every single protein, alright, the very first amino acid has a free amino group. Alright, so you always go from your amino group to your carboxyl group. 1 to whatever number the last amino acid is to make up your protein. Now, look here. You have one amino acid here and the second one. Alright? Now, notice they're actually held together by a covalent bond. Alright? Referred to as a peptide bond. Alright, so amino acids are strung together by a covalent bond called a peptide bond. Now, again, coming back to the idea of synthesis of biological molecules. Alright, so here, your pre-existing peptide has a free OH group. Your new little amino acid here has a free hydrogen as amino group. Alright, so you can basically pluck off a molecule of water and form your peptide bond. Alright, so this is a dehydration synthesis. Alright, so notice here, the very first amino acid, you have a free N-terminus. The last amino acid, you have a free carboxyl. Alright, so that's what's meant by N-terminus, C-terminus. Alright, your first amino acid and your last. Notice that the backbone of your protein, alright, is basically the framework, the skeleton that every single amino acid has. Alright, the side chains are what vary. Alright, your side chains can be polar and non-polar and charged. Alright, so this is showing you a representation of a much longer protein. Alright, so here, the very first amino acid, alright, has a free N-terminus, or a free amino group. Alright, at the very last amino acid has a free carboxyl group. Alright. And then you can just label these basically amino acid 1, 2, 3, 4, 5, 6, 7, 8. Alright, this shows you the first amino acid, free-hanging amino group, the last amino acid, free-hanging carboxyl group. Alright, so you always read it from N-terminus to C-terminus when you're talking about protein structure. Now, since your peptide bonds are covalent bonds, that means that since they're single covalent bonds, all right, that means that the molecules themselves, all right, that chemical bond can actually rotate on a three-dimensional axis. So that means the protein itself can change and alter its overall configuration shape, all right, which is going to be important a little bit later on, all right, because we're going to talk about the different levels of protein structure. All right, but right now understand that your peptide bond can bend and flex and rotate so that your overall amino acid can change its shape. Now it's going to change its shape to do one thing. All right, so we're talking about proteins, and a lot of times when we talk about proteins, we talk about proteins inside of a cell or being expelled outside of the cell into an aqueous environment. And so, since these proteins are going to be released into an aqueous environment, that means that your polar and your charged amino acids are going to be hydrophilic. Your nonpolar amino acids are going to be hydrophobic. All right, so what does that mean about the overall protein structure? All right, well, what that means is that the nonpolar amino acids try to get as far away from any water as possible. All right, and your polar charged amino acids try to associate themselves with water. All right, so this is going to affect how the protein folds in on itself. All right, so if you're a protein, you're stuck in a solution of water. All right, so if we have, for instance, here a beaker, and we get water, all right, and we got a little protein. Right now, I'm just going to draw, all right, this is a little protein. That means that out here, all these little lines, they represent your, they're pointing to your polar and your charged amino acids because they're polar or they're charged and they're hydrophilic. They interact with water. But your nonpolar amino acids, all right, I'm doing these little squiggles here, and the protein, all right, are in the middle of the protein. All right, they're trying to get as far away from the water as possible. All right, because they are non-polar. All right, so this is going to be important later on when we start talking about proteins in our three-dimensional shape. Your polar and charged amino acids tend to stick to the exterior, and your nonpolar tend to stick to the interior of a protein. Now, that isn't always the case, all right? And we'll talk about some exceptions later on, all right? But if you're a little protein, you're stuck in an aqueous solution, and you're just kind of freely floating around, all right? You're probably going to have a similar configuration to this, where your polar and charged amino acids are stuck to the outside, and your nonpolar amino acids are on the inside. All right, so again, we're going to come back to this idea of structure and function. All right, and the structure of a protein, its three-dimensional structure, its overall shape, is related to its function. All right. All right, so here you have a nice little ribbon and space filling model. And basically this is showing you that this protein there's a little indentation. There's a little groove here. All right. Now, this little groove is actually a binding site. All right. It's a site of biological activity. All right. So there's some target molecule here that's going to bind to this groove. All right. And only target molecules that look like this target and are chemically similar to this target molecule actually fit into that groove. And notice here, you only have a small part of that protein that actually fits in the groove here. All right. So this is really important when you start talking about like pharmaceutical drugs, or you start talking about toxins or poisons or antibiotics, because basically your drug is targeting some biological structure. All right, more than likely it's a protein, and so there's a small part of that drug that's going to interact with the target protein in a cell. All right, so later on we'll talk about enzymes. All right, but proteins have what we call active sites, which is this here. This is basically an active site. This is a site of biological activity. This is where your target binds to its what we call target molecule. Basically, the substrate binds to its protein. All right. So, looking at an example of structure and function. All right, so antibodies. All right, so antibodies are a great example. All right, so antibodies being proteins, all right, they target foreign substances. All right, so here you're looking at a portion of the flu virus. All right, so you're just looking at one protein from the flu virus, and you're actually only looking at a small part of that protein that is actually complementary to the antibody. All right, so this portion of the antibody recognizes this portion of the virus. All right, so this is how our immune system detects foreign bodies. All right, because this antibody is produced to target some type of foreign substance, some specific foreign substance. In this case, specifically this flu virus. And specifically this protein of this flu virus. And specifically this portion of the protein of this flu virus. Alright, so proteins have different functions. Alright, so we'll talk about proteins functioning as enzymes in a little bit. Alright, so enzymes are your basically workhorses. Alright, they drive chemical reactions. Alright, so chemical reactions can take place on their own. Alright, some occur very slow, some occur very fast. Enzymes help kind of expedite this process. Alright, defensive proteins like antibodies, alright, which we talked about in the previous slide. You have storage proteins. Alright, so proteins are a good source of energy. Alright, so think about it. That little baby chick in the egg. Alright, it's got ovoalbumin in it. Now the ovoalbumin's protein. And that protein, the purpose of that protein is it's basically a nutrient resource for that developing embryo. And so that ovoalbumin protein can be broken down into its component amino acids for the baby embryo to use for energy or for building other proteins. Alright, you also have proteins that are involved in transport. Alright, so when we start talking about transport systems later on in cell physiology, alright, you have proteins that are involved in transporting specific biological molecules, either into the cell or out of the cell. You have proteins that function as hormones, all right, so insulin, all right. This helps regulate the level of blood sugar. All right, you have proteins that function as receptors. All right, so proteins that stick out on the surface of the cell and that bind to chemical messengers, signaling molecules. So here you're shown an axon, all right, at the very end of a nerve ending. All right, so this axon is attached to another biological structure. It could be like a muscle cell. All right, and basically you have the release of a chemical messenger, something like acetylcholine, for instance. And this could actually be, for instance, skeletal muscle. All right. And the release of this acetylcholine, which is a neurotransmitter, it binds to its receptor. All right, so you have cholinergic receptors that bind to acetylcholine. And upon binding, it triggers a nerve signal into the muscle cell to basically contract. All right, so you have receptors, you have communication, you have proteins that are involved in movement. All right, so we'll talk about actin and myosin. All right, so muscle contraction. Talk about cilia and flagella, so the ability of cells to move through an environment. And you have structural proteins. All right, so structural proteins would be like those of keratin. All right, so the protein found in hair and nails. All right, so it's hard. It's rigid. It's protective. All right, on the surface of your skin. All right, skin cells are actually keratinized, and this actually prevents or aids in preventing water loss. All right. You also have collagen, which is a component of connective tissue. All right, so look at this collagen molecule. All right, it actually consists of 3 individual proteins that are intertwined. All right, and it's kind of like a rope. It can actually relax and constrict, and relax and constrict. And this is what gives it its elasticity in terms of making up your connective tissue. All right, that stuff that is attached to other organs of the body. All right, that very fine white mesh, if you've ever done a dissection, that's connective tissue.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells. The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction. Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos. Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals in animal cells to take in and use sugar. The hormone receptor is also a protein. Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins. Contractile proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal. Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins. Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call for a response. Gene regulatory proteins bind to DNA in particular locations and control whether or not certain genes will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings.
Alright, so next, we're going to talk about the different levels of protein structures. Alright, so this kind of gives you an overview. Alright, show how interconnected this process is. Alright, so we'll start off with primary structure, and then we'll end with the quaternary structure of a protein. Alright, now, all proteins have primary, secondary, and tertiary structure. Alright, there are others that have all four. Alright, but every single protein at least has the first three levels of protein structure. Alright, so first, kind of giving an overview of protein structure.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Alright, so first looking at primary structure.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Alright, so looking at primary structure. Alright, so here, alright, the primary structure is the sequence of amino acids in a protein. Alright, the order of each amino acid. Alright, so whether or not that first amino acid is a lysine or a glycine or methionine is going to determine, alright, the overall sequence. Alright, so now what determines the primary sequence of any given protein? Alright, well, that's tied back to the gene. Alright, the gene determines the sequence of amino acids. Alright, so the order of amino acids is actually encoded within the sequence of nucleotides. in the DNA of a given gene.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Alright, so this kind of gives you a picture of an alpha helix. Notice how it's kind of coiled like a corkscrew. Alright, hence the name alpha helices. Alright, so this is showing you the alpha helices without the side chains. Alright, so this is just showing you the ribbon model, the coils. Alright, so here's a beta pleated sheet. Alright, notice how it's flat and it's planar. And two strands of amino acids are actually anti-parallel to one another. Alright, so this is showing you more of a line image as opposed to the ribbon model. Alright, so when you're looking at secondary structure, alright, so you're talking about within that sequence of amino acids, you're talking about the amino acids interacting together within that protein. And you actually had the formation of hydrogen bonds. Alright, so like here, you have this oxygen that was a part of the carboxyl group of this amino acid. Alright, that's electronegative. It can hydrogen bond with this hydrogen that's attached to the nitrogen of this amino acid here. Alright, so because the nitrogen is electronegative, hydrogen's partially electropositive. The oxygen's highly electronegative. You can have the formation of a hydrogen bond, because that's what a hydrogen bond is. You have some highly electronegative atom interacting with hydrogen that's bound to another highly electronegative atom. Alright, so you have the same thing going on here. You have an oxygen, it's highly electronegative, hydrogen is partially electropositive attached to a nitrogen. You have hydrogen bonding. Alright, so the hydrogen bonding within the amino acids allow for the formation within a protein. You have regions that will have can have, they don't always have, but they can have alpha helices and they can have beta pleated sheets. Alright, so this contributes to the secondary structure of the protein.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Alright, so the tertiary structure. Alright, so this has to do with the interaction of the R groups. Alright, so recall, the R groups are either going to be polar, nonpolar, acidic, right, where they have a negative charge, or they're going to be basic, where they have a positive charge. Alright, and it's the interaction between R groups that gives rise to the overall 3D shape of a protein. Alright, so this kind of gives you an overview of the different types of interactions between your, remember, R groups for tertiary structure. Alright, so here, looking at hydrogen bonding. Alright, so you're talking about, in this case, this R group is going to be polar. Alright, and you have this highly electronegative oxygen interacting with this hydrogen attached to this R group here. Alright, so this example of hydrogen bonding. Here, alright, you have basically CH3 from one R group and CH3 from another R group. Alright, both of these are nonpolar because they're nothing more than hydrocarbons. Alright, they're nonpolar amino acids. Alright, so they can associate with one another through hydrophobic interactions. Alright, here you have two cysteine amino acids. Now, cysteine is really cool because it has sulfur as a component of its R group. You can actually have these two sulfur atoms from two different cysteine amino acids form a bridge, a disulfide bridge, which is a covalent bond. And then you can also have your acidic Alright, you could have your acidic and your basic amino acids. Alright, remember these have full charges. Alright, so they interact through ionic bonding. Alright, so this is what holds together your protein into this three-dimensional shape. Alright, so to give you a little bit better image of this. Alright, so you have hydrogen bonding between, for instance, oxygen and hydrogen attached to another highly electronegative atom like oxygen. Alright, so you have hydrogen bonding. You have hydrogen bonding again, in this case, hydrogen and nitrogen. Notice here that you have your alpha helices. Alright, so this is your secondary structure here. Alright, that's secondary structure. Notice that your overall protein, its shape, is determined by its tertiary structure. Alright, so here you have hydrophobic interactions, disulfide bridges, and ionic bonds. Alright, so you can see that the protein is starting to change and alter its shape. Different parts of that protein are beginning to interact with one another. And so what you end up with is a three-dimensional structure. Alright, so here you have a protein that has an overall three-dimensional shape. What is mainly composed of a secondary structure of alpha helices. Here you have a protein that has an overall three-dimensional shape that is mostly made up of beta helices. You can see some alpha helices at the top, alright, but it's mainly beta pleated sheets. And you have those that are kind of a mixture. Alright, you have beta pleated sheets and alpha helices. Alright, but the 3D structure is determined by the R groups and their interactions.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Alright, so again, every single protein is going to have tertiary structure. Alright, so that means that you have only one polypeptide. Now, a lot of proteins are actually kind of an amalgam of more than one polypeptide. Alright, so basically they have more complex structures. Alright, so they're made up of at least 2 or more polypeptides. Alright, so here, alright, talked about collagen. Alright, notice here you have a red polypeptide, a green polypeptide, and a blue. Alright, so 3 different polypeptides. They're all the same, alright, but there's 3 of them that intertwine to form the protein collagen. Alright, so the individual polypeptides have their own tertiary structure, but when they're combined together, alright, it gives the protein its quaternary structure. Alright, here, looking at hemoglobin. Alright, hemoglobin has 2 beta and 2 alpha subunits. Alright, so that means that it has 4 subunits that make up the hemoglobin protein. Hemoglobin as a protein has quaternary structure, but each of these individual alpha and beta subunits have their own tertiary structure. Alright, so this kind of gives you a walkthrough of looking at the different levels of protein structure. It tells you a description of each of those levels of protein structure and what actually holds them together. Now, when you're talking about quaternary structure, alright, the same chemical reactions, same chemical interactions that hold together proteins in their tertiary structure hold together polypeptides in their quaternary structure. Alright, so individual subunits are held together through hydrogen bonds, through covalent bonds, through ionic bonds, through hydrophilic and hydrophobic interactions, through van der Waals. Now, I mentioned how the genetic code determines the sequence of amino acids in a protein. Now, what happens if you alter that genetic code in some way? How does that affect the overall protein structure? What if you only change one protein? Does that really matter? I mean, is that really going to affect anything? Well, in this case, to kind of give you an example of how it does, how it really does matter. Looking at sickle cell disease. Alright, so you have one single amino acid that's been swapped out for another. And it has detrimental effects on the hemoglobin protein as a whole. Alright, so here you're looking at the different levels of protein structure of hemoglobin. Alright, specifically you're looking at the beta subunit. Alright, so here you have your primary structure of your normal hemoglobin molecule. You notice that the 6th amino acid is a glutamate. But in your sickle cell anemia protein, it's actually a phthalate. Alright, so you have a base substitution, or amino acid substitution. Alright, And so look at the secondary tertiary structure of the beta subunit, that protein, that polypeptide. Notice how they're strikingly different. Alright, this is more cylindrical. And this one actually has kind of a little pocket. Now, how this hemoglobin molecule is put together, how the beta subunits interact with the other beta and alpha subunits differs now. All because you've altered the one single amino acid in a structure. Now, these hemoglobin molecules, they don't interact with one another. Alright? They stay away. They transport oxygen like they're supposed to. Alright? Now, in your sickle cell hemoglobin molecules, notice how the beta subunits, you have these little binding pockets here, these little clefts. They now allow for the binding of other abnormal beta subunit proteins to interact with one another. Alright, so you get the formation of this like cylinder structure, so it aggregates to form a fiber. This is actually what allows for the formation of this kind of crescent moon shape to your sickle cell red blood cell. Alright, so these red blood cells don't transport oxygen as efficiently. You also have other complications that arise due to the actual physical structure of that red blood cell in terms of the flow of fluid through the vasculature, the flow of red blood cells and other elements through the bloodstream. Alright, so we talked about how the genetic code affects protein structure. Alright, but what about other physical, environmental, chemical factors? How does that affect protein structure? Well, what about pH? What about salt? What about temperature? What does that do to the overall protein and how it functions? Well, when a protein loses its ability to function, when the structure of that protein has basically unraveled, it's become denatured. And a denatured protein is an inactive protein. Alright, so here you're being shown an image of a protein that has its normal three-dimensional shape. So you can see the disulfide bonds and the hydrogen bonds that are holding together this protein in its 3D shape. Alright? Now, let's say you add a denaturant, a chemical. And what you do is you break all these chemical bonds that hold this protein together. Alright, so you break all the chemical bonds. Alright, and the protein basically unravels. Alright, when it's unraveled, it's no longer active. Alright, because a protein is only active in its three-dimensional shape. Alright, recall that proteins have different functions. Well, the function of that protein is completely reliant on the shape of that protein. And the shape of that protein is reliant on the interactions between R groups that hold that protein together. Alright, so a simple way to think about proteins and denatured proteins is think about an egg. Now, when you cook an egg, initially the egg white is clear, and then you have the yolk. Now, over time, that egg white becomes white and stiff. What's happening? Well, that albumin, the protein, is causing it to be white. It actually begins to denature. It begins to uncoil. And eventually it'll cross-link. Alright, because you can have interactions now between protein portions of a protein that were no longer previously accessible. Alright, so you now have cross-linking, which causes it to kind of solidify and become rubbery and harden. Alright, so a egg that's cooking is an example of a process of protein denaturation. Alright, this protein, this egg white, is never going to go back to its natural state. Alright, it is irreversibly denatured. Now, there are other ways to denature proteins. Alright, so you can use temperature. Alright, so you can boil an egg. Alright, you denature the proteins. You can use pressure. You can use pH. Alright, so think about it. We pickle food products all the time. We pickle vegetables. We pickle eggs. We pickle pig's feet. Alright, all we're doing is denaturing proteins by using substances that alter the pH. Alright, so one of the major components of, for instance, pickling pickles, cucumbers. Alright, vinegar. Alright, you have vinegar, you have salt. Alright, you have acidity. You have pickling spices. Alright, it's going to denature in the process of pickling that substance. You're denaturing proteins. You can add some type of denaturant, some chemical that denatures proteins, or you can use a mechanical process. Alright, so think about it. You take an egg white and separate it from the yolk, and then you physically beat that egg white with a whisk until it puffs up into a cloud. Well, you have physically, mechanically denatured that albumin protein. Another application of this is, think about it, hairstylist. Alright, the hair. Alright, so you have two keratin chains in the hair. Alright, so these two keratin chains are held together by disulfide bonds. Now, if you break the disulfide bonds through heat and chemicals, alright, you can wrap the hair around curlers. You can reshape the hair, and then as the hair cools back down, you have new chemical bonds that hold the hair together in a new configuration. Alright, so styling your hair is an example of denaturing proteins, and then allowing those denatured proteins to assume a new configuration, a new shape. Now, proteins. Sometimes they don't always want to fold like they should. Alright, so cells of our body. There are proteins that function as chaperones. Alright, so think about a chaperone on a date. Alright, what do? They make sure that that couple gets to where they're supposed to go and they're behaving as they should, right? Well, that's what a chaperone protein does. It makes sure that that newly synthesized protein, that it folds the way it's supposed to, it has the right configuration, right shape, and it gets to where it's supposed to be going.
In vitro, a denatured protein can refold into its native state on its own. Nonetheless, in vivo protein folding is aided by chaperones in order to increase efficiency and prevent aggregation of misfolded proteins. Molecular chaperones, such as HSP70, assume an open form when bound to ATP. In this form, the chaperones bind nascent polypeptide chains as they're synthesized on ribosomes. The bound ATP is hydrolyzed to ADP. ADP exchange with a new ATP molecule causes the chaperones to switch to a closed conformation, releasing the target protein. A second class of chaperones, the chaperonins, is required to help a small proportion of proteins fold properly. A partially folded or misfolded protein is inserted into the cavity of the chaperonin, where it can fold into its native conformation. ATP binding causes the chaperonin to expand and release the protein properly folded.
Alright, so your chaperone proteins. Alright, so here you have the chaperone protein with a cap, protein cap, and this hollow cylinder, alright, with a basically hollow tube running through it. And you have your polypeptide that's recently been synthesized, or let's say a protein that has been denatured, alright, can enter the chaperone protein. And inside this little cylinder, you have this little microenvironment that facilitates the folding or refolding this protein into its proper shape. And then once it's assumed its proper configuration, it's released. Alright, so there are all different types of chaperonin proteins in a cell. Now, enzymes are also a type of protein. Alright, so enzymes function as biological catalysts. Alright, they speed up chemical reactions. Alright, so here, alright, to give you an example. Alright, so you have, for instance, the sugar sucrose. Alright, sucrose being a disaccharide is composed of two monomers, alright, glucose and fructose. Alright, and they're held together by a glycosidic covalent bond. Now, this sucrose molecule will bind to an enzyme, a specific enzyme called sucrase. Alright, notice how the shape of the substrate, alright, the sugar, binds to the little binding site of the enzyme. Alright, this is referred to as the active site. Now, the reason it's called the active site is because this is where the biochemical reactions take place. This is where the biological activity takes place for this process. Alright? Your enzyme basically brings together reactants that are involved in a chemical process. It helps facilitate a reaction to move forward. Alright, so here you're dealing with a disaccharide. So when that substrate molecule binds, it'll actually cause the enzyme itself to change its shape slightly, so that it kind of much more tightly wraps around its substrate. And what it does is it puts a strain on that chemical bond, so that water can now come in and break that bond, separating this disaccharide into two individual monosaccharides, glucose and fructose. And now, once that process has taken place, the products are released and the binding site is open. Alright, so enzymes facilitate reactions. Alright, but they are not reactants, nor are they products. They just help to bring together reactant molecules, and they help facilitate the process of a reaction.